Well, we set the computer. I would like to say that it's a pleasure to come back and to meet old friends and actually make new acquaintances. So I'm actually really very happy to be here. Let me see if I set the presentation. Not there yet. Yeah, okay, very good. So I'd like to start by actually introducing my group. It is made by 6 people, so we have a small group at this time. So this is Shansé, this is Anki, Carmen, Kawai, and Emilia. And this is the place, Stockholm, the place that has kept me for such a long time. And this is down here, so one of the new installations that we have in the institute. So we have moved to a new building, and it's all brand new. Today I'll be talking about Parkinson's disease, and thanks to the previous speaker, I don't have to introduce much of Parkinson's disease at all. And what I would like to do is to focus the question that we have been addressing for many years, and it's the fact that the current therapies that are used for Parkinson's disease do not change the course of the disease. And we think that there is a need for cell replacement therapy because by the time that diagnosis is made, it's a huge loss of dopamine neurons that have taken place. So this is a cartoon showing some of the different strategies that have been developing over the years. We have here highlighted as number 1 is the first option, the first cell replacement therapy that was performed, which is mostly the work of Anders Björklund and Ole Lindvall. And what they did is actually use human fetal tissue for transplantation. So there have been different clinical trials, and it has been actually quite controversial because there have been many different results. But I think that from those trials, one thing one could learn is the fact that if you transplant cells that are prepared in the correct way, you can get good survival of the cells. And patients that have been transplanted with fetal tissue have been without medication, in one case up to 30 years. So this means that this is a proof of principle, actually, that cell replacement therapy could work with these patients. But of course, one of the main problems is that fetal tissue is very difficult to obtain. It's heterogeneous. It's very difficult to get a good quality control. So here is when the stem cell era started, and the option number 2, where we started to see that you can use other cell types like induced pluripotent stem cells that you can get from a fibroblast through reprogramming, or human embryonic stem cells. A third option that has evolved during the last year is with the idea of reprogramming. If you can take a fibroblast into an embryonic stem cells, perhaps you can actually take a fibroblast into a dopaminergic neuron, and that has also been proven to be successful. You can use it for transplantation, and you could have some effects on Parkinson models. And the last one that we have been involved in the last years is the direct in vivo reprogramming. In this strategy, we don't transplant any cell at all, but what we do is actually introduce the reprogramming factors directly in the brain and convert the cells locally in the brain into dopaminergic neurons. So all of these strategies, what they have in common is that you need to understand midbrain development, and only if you understand that carefully, you can actually mimic all the steps in the generation of dopaminergic neurons. So in this talk, I will be discussing 4 different aspects. One is a little bit of our studies on the generation of dopaminergic neurons, how they are made during development, and that's mostly our work with single-cell RNA sequencing. In the second part, I will discuss some of how can you apply this single-cell sequencing data to improve stem cell preparations for transplantation. In the third one, I will discuss our work on reprogramming, what we have here as option number 4. And then I will end up with some very recent work related to Parkinson's disease and genetics. So we start with the development. Here is the project that we started now a few years ago in collaboration with Sten Linnarsson and Roger Barker. And the idea was to compare by single-cell RNA sequencing the mouse and the human brain. One of our concerns was, of course, that despite the similarities between these 2 different species, we were worried that there might be some differences that could be important for midbrain dopaminergic neuron development. So what we did is take this single-cell sequencing approach, and the idea was to try to identify new cell types, new genes that are expressed that could be markers or could be critical for making dopaminergic neurons, and then, as I said, compare the 2 species. And this is the work of Daniel Gilbert, who was a PhD student at the time in my lab, and Joelle Lamano, also a PhD student in the group of Sten Linnarsson, that now actually has his own group in Switzerland. So this is what we learn about development. So this is a Disney plot in which we can see every single dot is a neuron, and they are grouped by similarity. So you can see, for instance, the dopaminergic lineage pointed out with these different arrows, going from progenitors to neuroblasts to the different types of dopaminergic neurons that we find in the mouse embryonic brain. We did the same type of experiments with human tissue, and what we see also is that during development, the process is similar. So we have also progenitors, neuroblasts, and then finally dopaminergic neurons. Now, what is interesting is that when we start looking at the molecular level at transcription factors, we started to see that there are differences. And I'm not going to go into detail in this slide. It's simply just to exemplify that. But I will show you one example. And this is the case of LMX1A, which is a transcription factor that is required for the specification of the dopaminergic neurons. If you knock out this gene, you don't get dopaminergic neurons. And what we see is that in the human brain, human development, this transcription factor, it's expressed in one radial glia. It's called radial glia 1 in human progenitors, and it's expressed very low levels in neuroblasts. And now when we look at mouse, we see that the picture is different. So radial glia does not express, progenitors express, and neuroblasts express at very high level. So if we think that this is one of the most critical factors in specifying or generating dopaminergic neurons, we realize that this transcription factor will coexist with other transcription factors and form transcriptional networks that are different in mouse and human. At least there will be cells in which this transcription factor will be interacting and networking with other cells. So one thing that we learned from this study is that if we want to make dopamine neurons for transplantation, or if we want to do good models of dopamine neurons for disease modeling or for drug development, we need to understand and we need to follow as much as possible human dopaminergic neuron development. And that brings me back to how do we use this learning in this case now in stem cell transplantation. So what can we do to improve cell preparations that will be used then later for transplantation in Parkinson's disease? And the idea is to use the single-cell RNA sequencing as a data. We know the standard curve of development. We know how development normally occurs. So does it happen the same way in our cells in vitro? And this was done by machine learning, in which you take the standards, you analyze, and then evaluate with that information the cells that you are producing in vitro. So now this is something that we published in this paper a few years ago. And what I'll show you here is that you have this wheel in which you have different cell types. I don't know if you pointer, yeah. So you have different cell types. In this polygon, in the vertex, you have the cell that is actually most close to the standard. And then as you go far away, so these are cells that are less good in terms of quality. So now we're thinking, this is normal development. So here we're showing at cells that are being generated. So you always have this tail. So you have the perfect cell and you have the tail. So cells that are being made and they are acquiring the appropriate gene expression profile. Now if we take these genes and cells and we randomize gene expression in the cells, we have this black cloud here in the center. So this is a random gene expression. So this is our negative control. So this is totally random. And this is how we see in normal development. Now what we did is analyze the human embryonic stem cells that were being used at the time. This is the protocol that was based in Lawrence Studer's in the publication is in 2011. And using that protocol, what we see is that there is a bunch of cells that are very close to the standards, to the progenitor cells, and they keep on generating these rivers of cells that go to the neuroblasts and then go down to the dopaminergic neurons. So in principle, we see the right process, but if you look at the two graphs, there is a difference, and it's that here the populations are very discrete. You don't find so many cells in between. So you actually find one cell, and then you find the other one. So there must be a rapid transition between one and the other. Whereas here we have a sort of a slow transition. If we look at the quality of the cells, and now it's looking one cell at the time, and we have a score. So the perfect cell is a score of 1. And we see in the human embryos, the dopaminergic cells are all close to 1, so they go slightly down until 60% of the genes that are alike the standard. But in the IPS clusters, in this case, we see that there is a very long tail. So this means that there are cells that express only 10%, 20%, 30%, etc. Again, supporting the idea that the cells are sort of coming along, but they do that in a not very regulated manner. And in addition, one of the things that we found is that there are serotonin neurons that are generated in vitro in these preparations. And that, of course, might be a problem because serotonin neurons have been suggested to be involved in the dyskinesias that are found in Parkinson's disease patients, and that were one of the problems in the clinical trials with fetal tissue transplantation. So the conclusions from this is that we have dopamine cells of good quality, but we have a constant generation, which makes that there are cells that are not as good as we would like in the preparation, and also that we have cells that are not the ones that we would like to be there, and those are the serotonin neurons. So obviously, this is, I think, probably we can call the second generation type of protocol. And what we have been doing is obviously start working on the third generation protocol. And in this third generation protocol, this is the work of Kaneyasu. So he's an extremely talented postdoc, and actually now he has his own group in Kyoto. So what we did, what Kaneyasu did is actually take some of the concepts that we have been developing along the years in terms of understanding midbrain development. And here I show you one of them, which is the use of laminin 511, extracellular matrix protein, to guide the development of dopamine neurons. We tend to think that the extracellular matrix is rather passive, but one of the things that we found is that laminin 511 promotes the expression of LMX1B, PATX3, which is a marker that is kind of hard to get in dopaminergic cells. And in addition, we have an increase in survival by this laminin. So what we found in this study is that in addition of that, one of the effects of laminin 511 is actually reduce the expression of hindbrain genes. So actually it is perfecting the midbrain character of the cells. Another thing that we have been studying for many years is actually Wnt signaling. And we know from the human development, this is immunostaining for Wnt1 and Wnt5. And in red, we have the dopaminergic neurons. And you can see that the dopaminergic neurons are completely surrounded by Wnt1 and Wnt5A protein. So this makes us think that the current protocol only uses activation of the canonical Wnt signaling, so that would be Wnt1. So we thought we need to incorporate Wnt5A to this protocol. And when we did that, what we found is that we have a better co-expression of markers, LMX1A and FOXA2, or LMX and OTX2. And in addition, we do have an increase in GRAIL1 when we use a high dose of Wnt canonical signaling activation. If we look at the final outcome, we see that we have a very nice curve of development, generating first LMX1A expression, then FOXA2, and that is actually followed then by the expression of NER1 1 week later, and then the tyrosine hydroxylase positive cells peaking at late stages. And these cells express PITX3, which is one of these markers that I told you that it's difficult to get. So this is all the work on the conventional markers, but of course what we wanted to do is look at the entire transcriptome of all of these cells and find out, are they really the right thing? So this is the work that we have been doing more lately, and actually this is the work of Kawai Lee, which is a PhD student in the lab. And what Kawai did is to use a very extended standard, and here you see that the wheel is much bigger than the one we were using before. And we include all the cell types in the ventral midbrain, and in addition, we include also 2 different cell types here, which are called BLMC, or vascular leptomeningeal cells. So this is a fibroblast-like cell that is found surrounding the vessels, and that it has been shown by the group of Malin Parmar and Thomas Pellman that it could be up to 40% of the cells, this group of cells here, this is also a Disney plot, 40% of the cells that are of embryonic stem cell origin in a transplantation. So we wanted to make sure that we wanted to test first whether we have these cells or not in our preparation. Obviously, we don't like the idea of having a cell that is not normally there in the ventral midbrain, because the proof of principle that you have a good outcome in terms of transplantation was done with fetal tissue. But now if we start introducing new cell types, we don't know how this is going to play. So this is our analysis. So now I make the wheels smaller, and this is looking at the differentiation of our cells from day 0, and so starting point, and day 11. And we see that there are two populations. One is the embryonic stem cell, and the other one, the larger one, is a progenitor cell type, which is called floor plate lateral. Now, if we go to 2 weeks later, so day 16 and day 21, we see that there is the emergence of two new populations. Well, first we see that the embryonic stem cells have disappeared, so this circle here, so we have a very good differentiation. There are no embryonic stem cells remaining. And the emergence then of two populations, which are neuroblast, in this case neuroblast lateral 1, and oculomotor neurons. And finally, by day 28, we see the emergence of a radial glia population, a neuroblast medial, which is the cell type that we know precedes the dopaminergic cell, and then the lower circle here to the right, which is the dopaminergic neurons. So we were actually very happy to see this flow, because we know from embryonic development, from characterizing it in fetal tissue, that this is actually a sequence of events. So by the time that we start looking at tissue at week 6, we see that there is a very large population of oculomotor neurons, whereas the dopaminergic neurons come just after. So here is the first one, and then these two other populations come. So, and we also see that the radial glia appears at this time the same as the neuroblast. So we think that our culture at day 28 resembles pretty good week 6 in the embryo. And of course, what is very important is that we don't have any of the leptomeningeal cells that are found in the other preparations is not present in our cultures. So this preparation mimics, I think, quite well what we know about midbrain development. This floor plate lateral progenitor is the one that expresses Wnt1, and immediately below is where the NER1 positive cells appear, which are one of the markers of this neuroblast mediolateral, and then the cells become dopaminergic neurons. So we think we are really getting with a protocol that mimics very closely, at least closer than ever before, the generation of dopaminergic neurons in vitro. So of course, after seeing this paper in bioRxiv, and also by interaction with the group of Malin, we were actually interested in finding out what is the composition of the cells that they intend to use in the different clinical trials. And here's a bit of a world map of some of the academic clinical trials that are in preparation. So actually the first one has been, or has already started, is the clinical trial of Jun Takahashi, in which they are using iPS cells differentiated into dopaminergic neurons. And we are very happy that we are going to be analyzing the cells that are being used for transplantation. So they keep an aliquot of every cell, every batch that they use for transplanting. We're also collaborating with a group of Malin Parmar, and they're coming with a clinical trial that seems to be, it will be the third one, in this case using human embryonic stem cells. And the second one will be that I think will be starting this year, is the one of Lawrence Tudor, and with whom we keep in contact, but I think they will be analyzing the cells themselves. So I think these studies are critical because since they are already happening, these trials, we are going to have a correlation of what is the molecular composition, cellular composition of the cells, of the human cells, and what is the clinical outcome. And we think that this will provide us a unique opportunity to actually be able to determine whether cells that may contaminate or may not be perfect, what is the impact and whether they are critical or not for transplantation. And of course, it's always very different to analyze one cell in vitro, as we are doing, to what is the cell performance and behavior in vivo. And at the end, the only way to really know what is actually happening is when you analyze the outcome. So this is the second part, the end of the second part, showing all the, let's say now, classical work on stem cell transplantation. And I would like to introduce you to the last part in terms of transplantation or cell replacement, which is the idea of now that we know that we can reprogram cells, why not using simply lentiviral vectors as we are using, or other type of vectors to introduce the reprogramming genes directly in the brain. And this is what Pia Rivetti has been doing or did actually in my lab for a while. And now she's actually in the group of Elena Cattaneo in Milano. So what we started is with this idea of reprogramming. Many people have been reprogramming fibroblasts into dopaminergic neurons, and that's the paper, for instance, of Caiazzo, 2011, and the combination of factors that they were using, LMX1A, a specification of dopaminergic neurons, ASCL1, which is required for reprogramming, and then they were using NER1. Now, in our case, we haven't used this combination, but we have preferred to use NeuroD1, which is one basic lipid gene that is expressed in the transition between progenitors and neuroblasts. And we thought that this was particularly important because the progenitors are the cells that are undergoing neurogenesis and then generating the neuroblast, which is a postmitotic cell. And in addition, we use a microRNA 218 because it's quite specifically expressed in dopaminergic neurons, and it was suggested to be involved in maturation. So this is the protocol that we have been using in vitro. So we have been using human astrocytes and reprogramming with these 4 factors that we call NEIL218. And we use a differentiation protocol that is rather similar to the one that we use with embryonic stem cells, with the difference that we also use valproic acid and the cytarabine. Now this is how the cells look like after reprogramming. So this is at day 13. And you see that they have nice neuronal morphologies, and you can see expression of markers of dopaminergic neurons, LDH1E1, GERK1, and dopamine transporter, etc. Now, these cells are actually more similar to the ventral midbrain dopaminergic cells that we found in normal development than to the astrocytes of origin. So this indicates that the cells have moved towards dopaminergic neurons. Still, there is a way to go, and I think we have a lot of work to improve this. We collaborated with a group of Tibor Harkany, and he actually helped us with some of the electrophysiology. And so here is these cells with multiple spikes and cyclic rectification, and this is actually found in about half of the cells. And we also tested this in vivo, so we actually use a transgenic mice in which we have the TTA transactivator under the GFAP promoter, and that makes that you have a transactivator only in astrocytes. So when you infect a construct that depends on the tetracycline, it will be activated, and in this case, it's only in astrocytes. So the idea is to see how the astrocytes that have the activator can actually turn on the expression of these genes, and then see the process of how astrocytes become dopaminergic neurons. And this is just a picture landscape of the ventral midbrain, of the striatal tissue in a cis-hydroxydopamine model. And just looking at 2 weeks after the reprogramming. And what we see, this is the control situation in which we have only GFAP positive cells, some very few TH positive cells. And in the case of the reprogramming, we see that there are lots of dopaminergic neurons. So we do get up to 1,200 dopaminergic neurons. So here we didn't do GFP in this case. Now, we have also used reporter mice to see, for instance, the expression of the dopamine transporter. And we see that there are the expression of markers in vivo. And we can also look at dopamine release. This is a fluorescent dopamine. And with KCl, we can depolarize that in slices from the striatum. And we also have electrophysiological properties in striatal slices as well. So with this, we thought that we could test this in vivo, and this is a work that was done with the group of Gilberto Fisone. And here I show you just part of this work in which we look at the pattern of walking of mice on a treadmill. And we see that in a cis-hydroxydopamine unilateral lesion model, what we see is that there is a decrease in the number of gait cycles that the animal does. So normally, there is an alternation of different limbs. And we see that the number of cycles decreases in a lesion, the same with GFP. And then with the reprogramming factors, we rescue the number of gait cycles. We also have a deficiency in the deletion and GFP in the quality of the cycles. So you have all these arrows in red that actually show different alternation of limbs, so that do not follow the normal pattern. And then we see that with the reprogramming factors, there is also an improving in the quality, and this is quantified here. So we have the alteration in the pattern of walking in the lesion, then in the GFP, which is about the same, and then the recovery with the reprogramming factors. And we have other parameters, limb alternation, and also asymmetry that is, again, rescued in the case of reprogramming, but not in the GFP condition. So we think that these results open up a new possible alternative in which one could maybe use reprogramming directly in the brain to replace the dopaminergic neurons. Of course, as it opens an opportunity, it also opens lots of questions, and we are fully aware that there are plenty of things to do. I mean, this work was done with lentivirus. We would like to be more gentle, for instance, use adenovirus. We have tested in a cis-hydroxydopamine model. We would like to use transgenic models. We have used it in a transgenic recipient. We would like to use it in a non-transgenic recipient. So I think we have a lot of work for the next years. One of the things that we have been doing is to try to improve the reprogramming itself. We have been using also the single-cell RNA sequencing data with regard to signaling pathways and extracellular matrix. So our new versions of reprogramming will include modifications there, as it will be also in the reprogramming factors. So we are investigating the networks that are formed by these factors. And we're also using bioinformatic tools to analyze that. And this is an example of a paper that we published now recently with a group of Antonio del Sol in Luxembourg. This part I would like to end it with showing you an example of a candidate approach that we followed from the beginning, and is actually trying to improve the basic loop helix transcription factors that, as I mentioned, are so important in neurogenesis. So here I'm showing you an entire Petri dish, so that's just completely scanned and stained with tyrosine hydroxylase in red. And we did this because it's always frustrating when you have these salt and pepper dishes. It's very difficult to convey the message, so we thought that the best way was to actually scan the whole dish so that you can get a proper impression of what is actually going on in a dish. So this is our combination of reprogramming factors, and this is how it looks if you take a view to some of these clusters here. And what we find typically is that there are about 16% of the cells of the astrocytes that are reprogrammed into dopaminergic neurons or induced dopaminergic neurons. We have used, for instance, this transcription factor, nascent helix loop helix, number 1, and this is expressed all along the dopaminergic lineage, but we didn't see any effect. But we have seen, for instance, all the transcription factor, TCF12. This is the basic helix loop helix as well. And in this case, we see that there is an increase in the numbers of dopaminergic neurons, but using others such as NeuroD6 or HES6 or even neurogenin2, we have a decrease in the numbers of cells or even a loss. So I think that here we have sort of a molecular code with basic loop helix genes that we really do not understand, because all of these genes that we are using here are expressed within the dopaminergic lineage. So we are wondering whether these combinations may actually be useful for specifying different subtypes of dopaminergic neurons. So I think we have a much more complex problem than what we initially thought we would have. But so this is the update until now, so at least we know that TCF12 is something that could actually help us to improve our reprogramming factor protocol. And now coming back to the original slide, so what I've shown you is that there are multiple ways of getting to cell replacement, and the idea is to go to Parkinson's disease patients. So the interesting thing of this last option that I just mentioned is that, of course, it would allow the possibility of doing a cell replacement without the need of cell banking, cell transplantation, cell preparation, GMP, et cetera, et cetera. So it may be an advantage. Of course, the disadvantage is that you do all the reprogramming directly in vivo, so if something goes wrong, then you have a problem there. But the idea is to try to perfect the method so that one could actually minimize this type of problems. So as I was saying, I would like to end up my presentation with very recent work that is also in bioRxiv, and is a collaboration with the group of Patrick Sullivan. And this is about the genetic predisposition to Parkinson's disease. So we know that Parkinson's disease is sporadic in 85% of the cases, but there are a lot of single nucleotide polymorphisms that have been identified in a sporadic Parkinson's disease. So there seems to be still some genetic component to Parkinson's disease. So the idea is that Parkinson's disease is emerging from the interaction of some genetic risk factors and also environmental risk factors. So we asked the question of whether we could identify which cell types are the ones that are at risk in Parkinson's disease. And the idea was to use all our knowledge of single-cell RNA sequencing and combine it with all the GWAS data that is currently available. And of course, as Jordi mentioned earlier, we have a very good situation because we actually collaborated with CIIIEN in analyzing and generating this gigantic map of the molecular architecture of the entire nervous system in the mouse. This is in adult mouse. And we started this collaboration with Patrick Sullivan and also Jens Jerling-Lefter. And what we did is collect all the cases that are available of Parkinson's disease. And also we entered a collaboration with 23andMe, who has been collecting data from many, many people. So we analyzed about 10 million different SNPs, and we found about 61 different genetic loci with about 266 annotated genes. And here is a picture of a Manhattan plot in which you see the different chromosomes and all the different loci which are marked by these dots going up. And if we do the annotation of these different loci, we can find many different genes. So here is not all the 266, it is only a selected list. But we see, for instance, that we have here GBA or synuclein or LARC2 as some of the top risk genes. But we have others that have not been described before, and one of them, for instance, that caught our attention is Win3. But there are many others, so there is a really huge variety of interesting genes. So what we did, as I was mentioning, is we tried to blend these two types of information. So all the risk genes, which normally are studied as a whole in one package, but now we try to distribute all these genes in which cell they are expressed. So now we attribute the risk of disease to a specific cell type using this whole entire nervous system map. And we did it in two different ways. So one is using a method that's called MACMA that takes into account all the genes that are expressed in the cells. And then the other is LDSC, which takes only the 10%, the top 10%. And so what we'll be looking here is at the most significant, which is the genes that are, or the cells that are significant with both methods in orange. And here I show you stroke, Parkinson, and schizophrenia. So schizophrenia, because it's actually one very good example of multifactorial genetic or risk or components. And stroke, because it's a very low predisposition. So we have here in vascular cells using one of the methods. And we see that the Parkinson falls in between. And when we look at these orange clusters, what we see is that the cells that pop up as risk of Parkinson are to some extent expected. So this is, for instance, the cluster that corresponds to dopaminergic and serotonin neurons. Here we have also some hindbrain cells. And the surprise came when we find that there is oligodendrocytes, several cell types in the oligodendrocyte lineage. And this is taking a close-up because this is, of course, all this huge amount of cells, so you cannot read anything here. But just blown up selected orange and some blues. So in Parkinson's disease, so we have dopaminergic neurons, so the cell that is called dopaminergic 2. In serotonin, we have the serotonin 2, 3, 5, and we have like 1 as well. And then different types in the oligodendrocyte lineage. So we have this committed progenitor type 1, but then we have a newly formed oligodendrocyte. So we have, well-represented population of cells. So we were actually, of course, concerned because there's nothing that is really been suggesting before that oligodendrocytes would be involved in Parkinson's disease. But of course, if one looks at the old literature, as the previous speaker also mentioned, you can see that, and also you see that I think he mentioned that there are these coil bodies that are present in Parkinson's disease, that you can find them actually in oligodendrocytes. So it's not that there is no alteration at all in oligodendrocytes. But anyway, so what we did is go this deeper, and one of the first things to do in this type of analysis is take all these different genes that are characterizing or are positive in every single cell type, and you eliminate them as a variable. So it's like sort of a bioinformatic knockout, if you want. And so here is, you have, again, all the different cell types. And what we are doing is, for instance, we focus on the oligodendrocytes. We are eliminating, I'll show you the magnification. We are eliminating the different variables, so this diagonal, so all the different genes that are typical of one of these cells. So for instance, here we have all the cells in the oligodendrocyte lineage, and you see that when we eliminate the genes that are in the oligodendrocytes type 1, we see that all of the other cells also lose the significance. So this means that the genes that we find in the oligodendrocyte lineage are shared by all of them. So they are not specific to that cell type. So they are specific in Parkinson, but not exclusive for this cell type. Now we can do the same, but now if you actually eliminate the genes that are expressed in oligodendrocytes, what happens then with the ones that are giving significance to the serotonin and the dopaminergic neurons? And we see that the significance still remains. So this means that there is two different setups of genes. So one in the oligodendrocytes and another one that characterizes the dopamine and serotonin neurons. But then we can ask the reverse question. So what if we now look at the dopaminergic and serotonin neurons and we bioinformatically eliminate the genes that characterize them, the GWAS genes that characterize them, and what would be the impact? So we see that in the case of the dopaminergic cells, when you eliminate the typical genes that are found in them that are associated to Parkinson's disease, you see that you lose also the significance for the serotonin, and when you eliminate the serotonin genes, you also lose the significance for the dopaminergic neurons. So this means that the dopamine and serotonin are very similar to one each other, so the risk is common for these two cell types. The risk chains are common. Now, what is the impact of this on the oligodendrocytes? So here it is, when you remove the risk in serotonin cells, you actually also lose the significance of the oligodendrocytes. So there is some common genes in the serotonin cells. And the same happens also with the dopaminergic cells. You lose in many of the cells, but there is one cell which is actually one of the earliest progenitor cells that actually the significance still remains. So from this analysis, one can conclude that there are two different sets of genes. One that is specific or more specific for the cells in the oligodendrocyte lineage, and others that are more in the dopaminergic neurons specifically. Now, this data we have also confirmed it using other sources of tissue. So, for instance, here we use as standards all the cells in development. So these are previous mapping experiments we have been doing. And again, we see that the dopaminergic and oligodendrocytes are also popping up as the most significant cell types. And when we use also single nuclei of postmortem samples, I mean, there is no midbrain sample available. It's something we are doing at the moment. But, and you look at different diseases, we see that in Parkinson's disease, we have clearly the oligodendrocytes popping up again as a significant cell type. So three different data sets giving us significance to the oligodendrocyte. And when we choose, we look at other tissue transcriptomic data that selecting now the 500 most differentially expressed gene in the substantia nigra, and then we plot these genes to dopaminergic cells across different BRAC stages. This is the data from this paper. We see that the oligodendrocytes are the first that are giving significance actually at the very early stage, BRAC1, BRAC2. And only, of course, in BRAC stage 3 or 4, we see the alteration in the dopaminergic neurons and later on. So we have also other studies in which we have also confirmed this data using not single cell, but in this case is full tissue transcriptomics. So I think this points in the direction that oligodendrocytes may hold some risk of Parkinson's disease, some genetic risk that has been previously passed unadverted. So to conclude, what I've shown you today is that the brain, the human brain, has a remarkable molecular diversity, and then single-cell RNA sequencing can be used together with bioinformatic tools to understand that and also to obtain critical information that could be used to stimulate neurogenesis or to develop new therapies. I've shown you also that it's possible to use direct in vivo reprogramming to actually change the fate of cells that are in the brain And of course, one needs to harness still all that potential. There is a lot of work to actually be able to selectively target your cells of interest, and also work to improve the methodologies. I've shown you also a risk of a genetic risk of Parkinson's disease that may lie in oligodendrocytes. And of course, we are now actively trying to understand that risk and model that risk in vitro. And finally, just as an outlook, I think that by understanding a little bit more of this genetic risk, we may actually be able to model disease in vitro, in this case, not genetic forms of Parkinson's disease, but perhaps also idiopathic risk of Parkinson's disease. If you saw in the GWAS data, some of the risk genes actually were as high as synuclein or any of the other genes. So there is a good chance that maybe with a couple of these genes, one could model, for instance, the oligodendrocyte alteration or possible contribution of oligodendrocytes. And of course, this may also bring us to a new strategies for developing therapies for Parkinson's disease. And I would like to end then thanking everybody that has been contributing to the work. I've introduced you before to the group, so Carmen, Chancey, Chris was not in the previous picture, Anki, Emilia, and Kawai. And then previous members of the group, Daniel Gilbert, that actually was doing all the single-cell RNA sequencing, Pia for the reprogramming in vivo, Kanayasu, the human embryonic stem cells, and Enrique, some of the early bioinformatic analysis. The groups of Stan Linarson, with whom we have been collaborating with all the single-cell sequencing work. Roger Barker, who has provided us with tissue to perform all this human analysis, and also some of the Parkinson work. Malin, Agnete, and Yun Takahashi for the collaboration on the human pluripotent stem cells that will be used for transplantation and analyzed by single cell. Marius Wernick for the reprogramming, and Patrick, Jens, and 23andMe for the genetic work. Antonio Del Sol, bioinformatics, and Gilberto, all the transplantation and behavior, and Tibor for the electrophysiology. And of course, the granting agencies without whom we also wouldn't be able to do our work. And thank you for your attention.
Thank you, Ernest. A very impressive presentation. Some questions?
Thank you, Ernest, for your beautiful talk. My question is related to the protocols and the transplantations in humans that are in progress, the clinical trials, or could be in progress. One specific question is whether the protocols are the same in Japan, in the Jun Takahashi, in Sweden, in Parmar, and in New York by Lawrence Tudor. Because, for instance, as you well know, the Jun Takahashi did a cell sorting after using extracellular molecules to drive the pluripotent stem cells to the dopaminergic neurons. I mean, at the progenitor cell stage, they did a purification. But for instance, you seem to have obtained a great protocol by 28 days. So are they, the 3 teams or 4 teams, are they using the same protocols? And the second question is, do you think that is needed to do, even in the best protocols, a further purification with cell sorting to further enrich the dopaminergic neurons or even get rid of the unwanted cells?
Yeah, so these are great questions. So all the protocols are what I call still second generation protocols. They are based on the idea of activating canonical Wnt signaling to increase the posteriorization of the progenitor identity. And all of these 3 have the same concept. Now, each of them has a slight variation. So, for instance, the group of Lawrence Tudor uses the chiron compound to activate this Wnt canonical signaling. Malin Parmar uses FGF8. Yun Takahashi, what he does is actually this cell sorting, as you mentioned, and also uses different extracellular matrix substrate. I think all of them are relatively similar. There will be some differences, and I think it will be very informative in the clinical trials. The one that is more different is also the one in Japan, because they use iPS cells instead of using human embryonic stem cells. The American is also, trial is also using H9 that have been sort of cleaned up and used now for clinical trials, and whereas the European trials uses cells that are real GMP from the beginning. So, I mean, there are differences, and it's difficult to tell how they will impact. Now, our protocol is very, very recent, so all these improvements have been done after the trials were launched, and they already have the cells, and they are already transplanting, and etc. So it's really too late for them to incorporate that. We think that obviously if that protocol that we are working on is an advantage, it may actually be used by them, and we would be very happy that they actually adopt it. But I think for the time being, we are not going to see that, so they are going to just go ahead with the trials as they are planned. And there was another question?
Basically, you answer. I mean, the need for cell sorting, but basically you answer.
Yeah, so in the case of the IPS, I think it may be more critical that you need this cell sorting, but in principle, it's not necessary. In our case, we also don't use any cell sorting, so if you instruct the cells correctly, you can, in principle, instruct exactly what you want. And this is our concept as well. So we're also working, for instance, in addition of this, to try to make a reverse engineering cell tissue for transplantation, where you actually build from the individual elements, so generating different reporters and then having all the different cells and be able to put together only the cells that you actually want in the transplant. So you could I think it would be the next generation of transplantation, so where you actually engineer exactly which cell types do you actually want to have in the transplant.
Thank you.
More questions?
Yes, about the surprising finding of oligodendrocytes being involved in the disease. Are there any alterations in myelin in the substantia nigra which have been found in the Parkinson's disease?
Yeah, so the main finding has been this that I mentioned, which is this deposits of synuclein in oligodendrocytes as coiled bodies. But to my knowledge, there is no clear implication of oligodendrocytes in the pathology. So it is something quite unexpected.
Okay, one more, and maybe we can go for lunch.
Thank you very much for your talk and your interesting result. I just was wondering how the dopaminergic cells that are implanted in the striatum integrates into a normal system of dopamine release, and especially the regulation of dopamine release that sometimes is fast and there are hot spots. So I'm wondering whether it has been studied, the different kind of cells that have been implanted and how they are regulated. Perhaps some of them they are regulated for dopamine release, depending on reward, motivation, etc. Could you comment on that, please?
Yeah. So the issue of hotspots is a serious issue, and this has been is related to the fact that if you put a lot of cells producing dopamine in one single place, of course you will have a very high level of dopamine and may be able to induce dyskinesias. What is happening with these late protocols is that the cells tend to integrate, and they disperse in the striatum, and they send processes. So when one does the tyrosine hydroxylase immunostaining, you actually see a rather homogeneous distribution. You always have a slightly more in the central core next to where the transplant is, but with time these cells tend to disperse. So this may be a factor, but we don't think that this is something that will be long-lasting as a problem. The other problem might be that, of course, the disease synuclein we've heard in the previous presentation is transmitted, so it can catch up with the cells that you are transplanting. And analysis of patients that have been transplanted with fetal tissue have shown that you see this transmission to the transplanted cells in around 12 years after the transplantation. But there was no sign of pathology in those cells, so meaning that we estimate that perhaps transplantation may be a solution for a window of time which might be in between 15 to 20 years, which, of course, for some patients it may be still a good solution. But of course, I mean, this is what I call the flat tire approach. So you have a flat tire, you take it out, put a new one, But still, we need to understand why. And the flat tire approach is very good because we don't have good remedies right now. So I think this is also why we are moving towards trying to understand really the mechanisms and the predisposition and how it does work, because in the long run, as we understand more about the disease, the flat tire will be less of a solution for disease. At the same time, the generation of cells in vitro are still good models for drug development in vitro. So we are not really married with any of these different solutions. We just want to see the field progress in terms of new therapies, and we would be equally happy if it is a drug or if it is a reprogramming or a transplantation approach.
The last.
Ernest, thank you very much for such a beautiful presentation. My question is related to the role of oligodendrocytes in Parkinsonian syndromes. There is a neurological disorder where oligodendrocytes are heavily involved, which is multiple system atrophy. I wonder if there are any studies on expression of RNAs or genes in oligodendrocytes in this disorder.
Yeah, thank you for the question. It's a very important point. So, of course, we're talking about Parkinson and synuclein deposition in neurons, but multiple system atrophy is actually the equivalent in which oligodendrocytes show that pathology. So, unfortunately, there are very few patients, and it's really hard to actually get sufficient GWAS data to make sense out of the genes, although some seem to be shared by both pathologies. So what we think is that we are talking here about a spectra of diseases, perhaps, in which we have on the one end Parkinson's disease, and on the other end we may have MSA. So I would love to have an answer for you, and it's something that we are very eagerly looking into the possibility of examining MSA and what might be the differences. But our explanation for the time being is that it may be a spectra of diseases, and that perhaps, we have perhaps too simplistically divided diseases in Parkinson's when it affects neurons and then when it affects oligodendrocytes. But, and that creates some sort of neglect when you look at one thing or another. But again, if one looks at the description of disease in nuclein, the coil bodies are found in oligodendrocytes from the beginning, from the first pathological description. So the point is that we actually neglect because of the classification. So Parkinson is neurons, MSA is oligos. So then you don't look at oligos anymore when you are working with Parkinson. So I think it might be related to that. Or there might be other explanations, but we are set to investigate that.
Okay, thank you very much. I think that if you have more questions, we can continue during the lunch. Thank you.